All right, chapter 2 is all about microscopy. We'll try not to get too bogged down in the physics of microscopy, but there are some kind of general terms and things that we need to understand to see our cells as best that we can. So we'll start with that. We're going to talk about some of the most popular kinds of microscopy out there and a little bit about staining. That's going to be some overlap with lab, of course, but I wanted to expose you to that, those of you that are not in lab. And again, if you are, it's good to hear something a couple different ways, hopefully. So, all right, so some principles that we need to understand in order to see our cells well through the microscope. So 3 things: magnification, resolution, and contrast. We need to be mindful of all 3 of those principles. Magnification is the obvious one, right? Apparent increase in size. And if we want to figure out the total magnification, we take the ocular lens and multiply that magnification times the objective lens to get the total magnification. Okay, so the ocular lenses, that's typically 10x. Should be printed somewhere on the microscope you're using, but that's most common. And then there's usually several different objective lenses. So for our scopes up in the lab, the ocular's 10. Say for the lowest power, that's a 4x. And so with that lowest power or the scanning objective in place, we get 40x for total magnification. Okay, so that's pretty easy, but we can, there's limits. The scopes that we have will magnify up to about 1,000 times. And pretty much we could magnify beyond that with a regular light microscope, but it doesn't really do us any good because at that point we start to be limited by resolution. Even though the objects that we're trying to see are bigger, the clarity is not good enough on them. Okay, so resolution takes a little bit more work to define. We're going to call it the ability to tell that 2 objects are separate. Okay, so this is not the same thing as being in focus. You're going to assume that you have your cells in focus. Okay, but it is kind of a measure of clarity. Basically, with our light microscopes, here's the cell wall of a cell, and here's the cell membrane, we don't have good enough resolution to tell that those are 2 separate objects. They look like the same thing. If we use a microscope, some kind of electron microscope that has better resolution, then we can see a cell wall and a cell membrane. So we can tell that there's a space in between there and that they're 2 separate objects. Okay. So if we wanted to kind of monkey around and get the best resolution that we can, we need to understand how we figure that out. The resolution, or sometimes we say resolving power, of any microscope is going to be equal to the wavelength of light divided by the microscope's numerical aperture. And actually there's a constant in here that we multiply by that actually we're not going to worry about, but just wanted to mention that. So wavelength of light divided by numerical aperture. First thing that you want to understand is resolving power. Good resolving power is going to be a low number because this is actually the distance between 2 objects where you can still tell that they're separate. So if the resolving power is really small, that means that the objects are really close together and you can still tell that they're separate. If they have to be really far apart in order to tell that they're separate, then that's not as good resolution. All right, so how can we manipulate this part of the equation to make this smaller? Okay, either we want the denominator to be smaller, sorry, numerator to be smaller, or we want the denominator to be larger. Okay, so play around with that, right? 1/4 is better resolution than 1/2. 1/4 is a smaller number than 1/2. Okay, or if the numerator is smaller, right? 1/2 is smaller than 2/2. Right? Okay, so we either want a smaller numerator, shorten the wavelength of light, or we want a larger numerical aperture. Let's talk about wavelength of light first. So here's a spectrum of energy. Visible light is anywhere wavelength between 400 and 700 nanometers. So by wavelength, I mean distance from a crest to a crest, or the distance from a trough to a trough. Okay, so visible light has certain wavelengths, and you know that red light has a longer wavelength than purple light because when you see a rainbow, the red is always on the outside and the purple is always on the inside. Okay, so that's visible light. Of course, we get longer wavelengths. We have infrared, radio waves, microwaves. Shorter wavelengths, which, by the way, have increasing energy as we get shorter and shorter. Wavelengths in the ultraviolet range, or UV range. We're going to talk more about that later because that kind of light is a mutagen. As well as X-rays, gamma rays, and even electrons, shorter and shorter. Okay, so if we can possibly make the wavelength of light shorter and shorter, that's going to give us a smaller numerator and give us better resolving power. Okay, so to improve resolution, we're either going to use a shorter wavelength. Okay, if we use electrons, which kind of act like a wave, much shorter wavelength than white light, which is going to be somewhere in this range. Okay, we're going to have much better resolution. Even if, say, we used a purple light rather than white light, in theory that would give us a little bit better resolution. I've never actually tried that, and I don't think that it would be enough to make much of a difference if we were trying to just manipulate our a bright field microscope like what we use in lab. Obviously, we can't use this microscope and use electrons instead. That wouldn't work. But if we used a blue lamp, maybe that would give us a little better resolution. The other thing that we can do is monkey with this. What is this numerical aperture? We're going to define this as a measure of the angles of light. I think I said angels. Angles, E-L-E-S, yep, sorry. Measure of the angles of light allowed or accepted into the lens. All right, so let me illustrate what I mean by numerical aperture and how we can monkey with that. Well, this involves something called refraction. So let me go on to this picture actually first. And you have experienced refraction if you've ever gone fishing and dipped the end of your fishing pole into the water and noticed how it looks like suddenly your fishing pole bends off at a weird angle. Okay, you've just experienced refraction. Basically, when light waves are traveling through a medium, when they change mediums, when they go from the air to the water, they appear to bend. Okay, so that's refraction. It's not really bending. But the same thing happens when light is traveling through different mediums in a microscope. I'm not crazy about this picture. Let's say this is maybe the 4x objective, because when you're using that scanning power objective, that's the shortest one. You have your light source underneath the stage, and when the light travels up, of course, your cells are going to be on your slide, probably right here, and the light's traveling up through the slide, and then it hits the air. That light is going to bend out in all different directions, okay? It's really going to refract a lot. But we can minimize that refraction if we use a special lens that is 100x, and it will say on there oil immersion lens, because that's a lens that's specifically designed. You put a drop of oil right on there where your cells are. It's kind of hard to see that. And then that oil has the same refractive index as glass. So when the light comes up through the slide, it's going to bend much less, and more of those angles of light are going to be captured within the lens. Okay, so you can kind of see that a little bit better here. You can kind of see the oil there. It's going to look like when you turn that objective into place, it's going to look like, oh, I'm going to scratch the slide. It's too close. No, it's designed to be that way. That 100x oil immersion lens will not focus if it's not down into oil. Your book says that you might be able to do it, and trust me, many students have tried, and just out of getting mixed up and not realizing which lens they had engaged. But that will not focus. You will not be able to see your cells unless you have some oil on there. The other lenses, the 4x, the 10x, and the 40x, are not designed to be used with oil. Now, it's not going to be a problem, especially that low power and the medium power. If there's oil on your slide, you'll still be able to focus just fine. But for that longer objective, that high power one, if you get oil in there, then it just junks it up and you're not going to be able to see clearly through there. So it doesn't normally, again, hurt it to have oil on it, but you don't want to get the oil actually in the lens. Okay, so it's fine to go back and forth from the low power to the oil immersion one, but you don't want to drag that 40x one through the oil. Okay, so what I'm getting at here is when we use a oil and an oil immersion lens, that's going to improve the resolution because it increases this numerical aperture. Okay, so use oil and a specially designed oil immersion lens. And that will improve the resolution. So when we're viewing our cells in the lab, we always we start out with low power just to find the cells, but then you're going to go right up to the oil immersion because your cells aren't going to look like much unless we have that good resolution. Okay. And the last thing that we need to think about is the contrast, and that's a little bit easier to wrap your head around. That's, we're going to define that as the ability of our cells to stand out from the background. Okay, so here's an example of some cells with contrast and some cells without contrast. These are cheek cells. You might have taken a look at your cheek cells in a Bio 120 lab. I'm not sure if they're still doing that or not. But here's some cheek cells that have very little contrast, very difficult to find, actually. And then here's some cheek cells where a stain has been added. So to improve that contrast, the first thing we think about doing is adding some kind of stain. So here it looks like they added a couple different stains, maybe one that makes the nucleus show up a little bit better. Incidentally, all these little dots, those are bacteria. So it gives you an idea of how much smaller the typical bacteria cell is compared to a typical eukaryotic cell. Okay, so to improve the contrast, often we're going to stain. Unfortunately, this will always kill the cells that we are staining. So that's a little bit of a drawback, but at least we can see them. If you really don't want to add stain, another possibility, sometimes you can be successful dimming the light. If you get that dimmed just right, then the edges of those cells will show up a little bit better, and you may find that you don't absolutely have to stain the cells, but it takes a little bit of practice to be able to do that. If you increase the light, that just bleaches everything out, so you want to lessen the light. Now, there are some different kinds of microscopy designed so that you don't have to stain cells in order to see them. So I'll talk about one of those in just a minute. All right, the different kinds of microscopy that we're going to cover, these are the most common ones used. There are at least 2 or 3 more different kinds that they talk about in your book. So if you're interested, you can read about those. These are the ones that I want you to know a little bit about and even be able to recognize some, what kind of microscopy that is if I show you a sample. Okay, so let's go through those. Obviously, a bright field microscope, that's going to be the most common kind of microscope used. These are the ones that we use in lab. Okay, so most common, we typically see a light background. Typically, we're going to stain cells. Okay, now it's possible to stain the background and not the cells, but assuming that you haven't done that, the background's going to be light. Typically, we can get our magnification up to about 1,000 times is the limit there, and a resolution of about 0.2 micrometers. Okay, so the parts of the scope, I think I included a couple of homework questions asking about the parts of the scope, but just kind of for practice, but primarily going to leave that to your lab instructor. Ocular lenses, like we said, objective lenses. Underneath the stage, there's going to be some condenser lenses and an iris diaphragm, which is just a little lever that's controls the amount of light that comes up through the stage. Totally okay to mess with that lever a little bit to get kind of the right amount of light. There's going to be a big knob, that's a coarse focus, that will move the stage up and down quite rapidly, and a fine focus that will move it in minute amounts. Okay, let's see, what else did I want to say about that? I guess that's pretty much it. Light microscopy. Very similar kind of microscope that also uses visible light is a dark field microscope, and the only difference is that between it and a bright field microscope is going to have a dark field stop that's covering the condenser, so kind of right down here somewhere underneath the stage. And that stop, here it's kind of turned up on the edge, on its edge. So if you laid that down flat, basically you have this dark in the middle here, and then the light is only going to be able to come up around the edge, so it's going to hit your specimen cells at kind of an oblique angle. So basically your cells are going to appear light against a dark background. The light kind of gets reflected off your specimen cells and make it appear to be emitting light, even though it's not really. Apparently the resolution is just maybe a little bit better, probably not by very much. Definitely gives us really good contrast. Okay, so that's the big advantage of a dark field microscope, is that staining is not necessary. Okay, so you can quite easily view living cells. So it's kind of fun to see them moving around. All right. All right, that's dark field microscope. Here's an example of if you compare dark field microscopy to bright field without staining. Again, it's kind of hard to see those cells there with bright field when you haven't stained. Dark field, dark background. Fluorescence microscopy also will have a dark background, but we always need to stain our cells here. This is not using visible light, but light in the ultraviolet range. So that's somewhere between about 100 and 300 nanometers. Okay. So remember, that's a shorter wavelength than visible light. So a shorter wavelength, that's going to, remember, improve resolution. Going to get better resolution than you would for a bright field or a dark field microscope. And it's going to use dyes that fluoresce. In other words, they become excited when the UV light hits them. And these microscopes are going to have a stop in the ocular, so you're not, shining an ultraviolet lamp in your eyes. You obviously wouldn't want to do that. Okay, so this can be a really powerful technique as a diagnostic. It's often been used to diagnose tuberculosis. If both samples here are of a taken of a sputum sample. So the sputum of a patient is stuff that's hawked up from your lungs. So not just spit, but stuff that's really coughed up from the lungs. And you can see if you look at a sputum sample for regular bright field microscopy here on the left, you just kind of see a bunch of junk in there, and it's kind of not easy to tell what's what. But if you use fluorescence microscopy and you use a special dye that's differentially sticks to a component in that bacterium cell wall. We have a fluorescent dye that sticks to mycolic acid, which is present in that Mycobacterium tuberculosis bacterium. And you can see here that the bacterium will fluoresce. And so we're here, it's there, you just can't see it. But now that we can see it, and that person has an active case of TB. Okay, so that's a powerful technique. Moving up to a couple different kinds of electron microscopy, huge advantage in terms of the magnification limits and resolution limits. Again, because the wavelength of electrons is much shorter than light, so we can get somewhere between 10,000 and 100,000 times magnification, even potentially up to 1,000,000 times magnification. So again, 10 to 100, maybe even more times magnification. Resolution is better. It depends on which kind of electron microscopy we use. Unfortunately, it's cumbersome in a couple ways. Number one, you're not going to have these in your dorm room. They're quite expensive. And the cells have to be fixed, in other words, prepared, and viewed in a vacuum. So in the absence, two U's, not two C's. Oops. Sorry, there we go. So in the absence of air, because air will scatter those electrons. So, excuse me, do that in a vacuum. All right, so this is a picture, I believe this is a transmission electron micrograph. Everything, you're not actually looking at the actual cells. It will show up on a computer screen, and the computer kind of constructs the image for you. All right, so Transmission electron microscope, a little bit more about that. Here's a couple of examples of what the image would look like. The image is going to be a cross section. Okay, so transmission, the electrons transmit through the cells. The cells are thinly sliced with a device called an ultra microtome. Oops. Again, spelling, no B there. Okay, so if you think, the machine that cuts your deli meat, okay, picture that. Only an ultra microtome can cut a red blood cell in 100 sections. Okay, so that's how thinly those slices need to be. And it's potentially pretty problematic. I've heard of researchers spending a month trying to get a good picture of their cells, to publish. It's quite a process to prepare the cells. They have to be dehydrated, embedded in plastic, and put on a copper grid. So sometimes the image that you see can be a little bit distorted, and it takes a lot of practice or some specialization to be able to understand exactly what you're looking at. The color here is artificially added, so it's always just going to be a black and white image. Sometimes then we'll add the color just to help people understand kind of what exactly that they're seeing. But it is a cross section. Okay, and this gives us the best resolution. Okay, so we said the resolution for a bright field microscope was 0.2 micrometers. Here we can get the resolution to about 0.3 nanometers. Okay, so that would be, if we converted that to micrometers, we'd move the decimal 3 points to the left, so that's the same thing as 0.003 micrometers. Okay, so smaller than what we get for a bright field image, so better resolution. Okay, the scanning image, and you can see down here TEM, right, transmission electron micrograph. The scanning images are often a lot more exciting to look at. These are not, for this kind of microscopy, you don't thinly slice your specimen. What we get is a 3D image of the specimen. So the electrons are not passing through. In order to prepare the specimens here, they're usually coated with some kind of really thin layer of some heavy metal, like gold. So the resolution is not quite as high as transmission electron microscopy. But again, still much better than what we get for using ultraviolet light or regular visible light. So these images, you can kind of actually tell what they are. A here is a marine diatom. B here is the spore structure of some kind of fungus. We have a paramecium, and here's some bacteria cells. So again, a 3D image. Okay, these are not cross sections. Occasionally, it's kind of hard to tell the difference, but again, if these cells were a cross section, you would see what's inside of the cells. Okay, so lastly, I want to talk a little bit about staining. Again, I know your lecture professor is going to talk about this more, but it's a good introduction to it and a good exposure to it if you're not in lab. So some principles of staining. Now, again, I'm talking about using a bright field microscope. We're not going to get into how to stain cells for electron or fluorescent microscopy. all the dyes that we use in lab are either going to be basic dyes or acidic dyes. And if they're a basic dye, that just means the colored portion of that chemical, that's the dye, it has a positive charge to it. And that's important because then that's going to adhere to the negatively charged bacterial cell wall. Okay, and all bacterial cells have a slight negative charge to their cell wall. Okay, we'll talk later about the difference between a gram-positive and a gram-negative cell, but that doesn't have anything to do with the charge of the cell wall. It's just positive for this gram reaction or negative for a gram reaction. Okay, so most of the dyes that we use, they're just going to stain the outside of the cell wall. There will be one case where we're going to use an acidic dye, and as you might expect, this is a dye that has a slight negative charge. So that's going to be repelled by the negatively charged cell wall, and it's going to stain the background. Okay, so either way we get good contrast, whether we stain the cells and not the background, or we stain the background and not the cells. When we do simple staining, that just means that we use one dye. We don't bother to do that in lab because it doesn't give us a lot of information. But you can see the cells, you can see the shape of the cells, how the cells arrange. Like here, these cells tend to arrange in clusters, it looks like. You can kind of tell how big they are, but that's pretty much, use one dye, that's what I meant to say there. That's pretty much all the information you get. If we do some kind of differential staining procedure, that means we're going to use 2 or more dyes. And that's going to give us a little bit information because different cells will stain a different color. That enables us to put some of our cells into groups or maybe see a particular structure better if we use 2 or more dyes. Okay, so here's your book's diagram here, or a little table of the differential staining methods that are out there. I'm pretty sure this is the 5 that are there. They're the only ones that I have ever heard of. We will do 4 of them in lab. Okay, so you're going to do lots of gram stains. We'll talk more about next week when we get to cell structure, about what makes a cell take up one color or the other. Some cells have a different kind of cell wall, and we call them acid-fast organisms, so we're going to do a staining procedure to see those. One that we can especially visualize, endospores. We'll talk more about those next week. The one that we don't do is the flagella stain. I've never actually done this one. I've heard it's fairly cumbersome. Excuse me, the reason we don't do it is because it doesn't work out very good sometimes. But we actually forget that most of the organisms that we stain in lab do have flagella. Those flagella just don't show up when we do a regular gram stain. We have to, again, use some special dyes to do that. The capsule stain is the one where we use the acidic dye in order to stain the background, because the capsules just don't show up very well when we try to stain the capsule itself. Okay, so the one, I'll close here just talking a little bit more detail about the gram staining procedure, because it's the first question we ask about an organism when we're trying to identify it. Is the organism gram positive or gram negative? And it's really just the difference between 2 kinds of cell walls. And again, we'll talk more about that. But, in the hospital, it makes a difference if your infection is caused by a gram positive and a gram negative, because that depends, we use different antibiotics based on what kind of organism it is. So basically, I'm not going to go through the gram stain in lecture. I'm going to make you learn that for lab if you're in lab. But it's a series of different stains that if the cell has a certain kind of cell wall that retains those stains really well, that first dye that we add to it is purple, and it holds on to that and is not decolorized very easy. Okay, so that purple cell, that's a gram-positive cell. The gram-negative cells have a different kind of cell wall that gets decolorized very easily, so it takes up, well, depending how you count it, the second or the third dye that we use, the counter stain, and that's going to be a gram-negative cell wall. Okay, so you'll be doing lots of that. Now that we've finished chapter 1, chapter 2, and 7.6, so you have some homework assignments that you can be working on. Thanks for bearing with me, and I hope to see you Wednesday.